Hi everyone, I'm Pranoti, host of Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials or nanoscience, who tweets from the Real Scientists Nano Twitter account, which is realsci_nano.
Hi everyone, my name is Pranoti. I am your host of Under the Microscope podcast series, and we are switching the format a little bit. So now you will get one long episode for every curator, every materials or nanoscientist who is a guest on this podcast. And today we have with us Jessica Henty-Ridilla, who is an assistant professor at the Upstate Medical University, SUNY, which is apparently not called SUNY, it's called SUNY, which stands for State University, New York. So, hi Jessica, welcome to Under the Microscope. How are you?
Good, thank you.
Awesome. Is it sunny in Sunny?
No, it's actually raining today.
Oh, okay. You got my joke, right?
Yes, that was great.
That was a good one, yeah? Okay, cool.
The sun always shines on SUNY campuses.
Right. Enough with the jokes. I think we have been too harsh on SUNY. And for those who are listening on our podcast, who are only listening to the podcast on Spotify, Google Podcasts, or Apple Podcasts, and all other kinds, do watch the YouTube video for this funny exchange between Jessica and I about how sunny it is in SUNY. How it is not sunny in SUNY. Okay, well, never mind. Let's get to the science. Jessica, could you please explain your research to us in super simple words, please?
Okay, so in my lab, we study the cell's cytoskeleton, which is specifically for us actin and microtubules. And these are like the bones of your cell. So we kind of joke in the lab that if you study the cytoskeleton, every day is a bones day. Combined, these two proteins, actin and actually tubulin, which is the protein that makes microtubules, actually dictate most of the things that a cell does, either its shape, how it moves around, how it divides. We are particularly interested in how this regulation goes wrong in neurodegenerative diseases, specifically amyotrophic lateral sclerosis or Lou Gehrig's disease. But it also gets misregulated in cancer and a lot of other disorders.
Okay, a lot of very complicated sounding diseases and cancer. Okay. Wait, so okay, this is like a completely new field for me. This is like biology. So a lot of the terms that you use, other than bones and cells and cancer, it was a bit too fast for me. So you study two proteins which are responsible for bones.
Yeah. So do you know what else? You could think of them as Legos. And one might be like the advanced Legos that you graduate to that are really like small pieces. Mm-hmm. And the other is like slightly bigger pieces. But basically, they're constantly being built, just like you can build with Legos. They can get added to one end for the most part. Mm-hmm. And there's proteins that help bring those things together. Mm-hmm. And there's proteins that disassemble and recycle them. And just like in like the Millennium Falcon Lego kit, there's only a couple shapes. And I mean, in the case of the cytoskeleton, there's like a couple shapes, but you can build these really elaborate structures from just those little tiny shapes. And that's basically what we look at. It's really beautiful. It's really pretty to look at.
That sounds so cool. But we are not talking about making bones, right? We are talking about
not real bones. Real bones. In a sense, there's cells that have a cytoskeleton that are part of your bones, but this is like if you think of how a skeleton holds your body up.
Yeah.
These are the proteins that hold your cells up.
Right. Okay. And every day is a bones day.
Yeah.
What was that? Sorry, I missed that part. I didn't.
So there was this cute internet meme. I don't know what's going on with it. And every day it was either a bones or a no bones day based on this really old pug that if he would sit up, then he was having a bones day. It was like his body had bones in it. And if he was having a bad day, he kind of like slumped over and it was like he didn't have any bones. It was really cute. And it was supposed to be like a day that you go and take care of yourself. It helped to promote like self-care and mental health awareness. It was really cute.
Oh, that is so cute. I completely missed this.
You should look it up. I think they still tweet every once in a while, but it's really great. It was cute.
That is so cool. So when things work in your lab, it's a bones day. When things don't work, it's a self-care day. Like, okay.
It's a no bones day and it's a self-care day. It means you should like maybe rest and regroup and get yourself back together and then try again the next day.
And hopefully the next day will be the bones day.
Yes.
Wow. Oh wow, this is so cool. Okay. So how did you become the bone doctor? Can I say that? No, how did you end up being the professor at SUNY, close to New York? So how did that happen? Tell us about your career journey so far. Bone doctor.
Yeah.
Bone doctor? No, probably not.
Okay, so basically we started kind of an interesting question about how I ended up where I am, right? How does a plant biologist end up at a med school? So that's what my story basically is. I started my career, as an undergrad. I was dissecting a mouse for the first time that was just like sacrificed and doing a DNA extraction, and my hand just kind of like grazed it. And I still felt its like warm little body. And it was just a really scary moment for me that I was like, you know what? I think I'm going to do plants. And I always loved plants. That was one of the things that I got really interested in science about. It turns out the plants have these proteins too. And I specifically studied how the actin proteins, which make these nice structures, get disassembled. And it turns out that they get disassembled during a lot of key moments, including when plants are attacked by pathogens. And that's really important because we have to eat. And if all of our crops get, attacked by pathogens, that's not very good. So this is one of the first things that pathogens can hijack to do whatever they want. And I thought that was really cool and really just fell in love with microscopes and this protein because it's really beautiful to look at. And I got to use a lot of fun microscopes, learned a little bit about biochemistry, but that's when I really fell in love with the cytoskeleton. And there's a lot of different proteins that technically make up the cytoskeleton. So I also started to get curious about these other proteins too. And when I went to do a postdoc, I wanted to learn more about biochemistry and this other protein, this other component, microtubules. And I sort of felt like all of the labs that were getting a lot of airtime at conferences weren't necessarily doing plant research, and plant research is really important. So I sort of wanted to change it up and try a different model system. And it turns out that neurons are a beautiful system for looking at both actin and microtubules at the same time and how those two things talk to each other. So I got interested in neurons. And I learned about that during my postdoc. And then we had some really great imaging, really cool stories about actin and microtubules talking to each other and how that coordination was going, specifically in neurons. And I was able to sort of appreciate that these things are more than just a beautiful architecture, that they have really specific functions. And when those beautiful architectures aren't totally intact, a lot of neurodegenerative diseases happen. And one of the regulation proteins that we're really interested in, profilin, actually can adjust how both actin and microtubules polymerize. And so there's mutations in that gene that also cause ALS. So in my own lab, I thought that would be the new direction where I could take some of the skills and tools that I'd learned about, but also, look in a more disease relevant way. So what's great is it doesn't matter what model system. A lot of proteins are the same, regardless of where you're studying them from. And some genetic systems are easier to use than others. And when you kind of, get inspired from other fields, I think it helps you get a different viewpoint that maybe hasn't totally been looked at before. So I'm really excited. That's kind of how we ended up where we are. And I don't know.
Wow. Oh, that's quite a journey. So you managed to take the best of both worlds, and I hope you don't have to work with mice or any other animals anymore to feel their warm body while you.
I think that that's really important. But for what we're interested in, we don't necessarily need to do that. And we can have a friend that's like an expert at doing that when we build it up in our immortalized cells now. And it's okay. So I think it's important, but it's not necessarily important for me to be doing that. So and as far as plants go, even though I have a PhD in plant biology, I still can't figure out how to keep office plants alive. So, there's that.
That will be such a relieving thing to hear for our listeners, because a lot of people think that they kill plants. I have lots of friends who are like, don't give me a plant. I can't take care of a plant. And yeah, even a plant biologist doesn't always figure out how to keep the plants alive. That's good.
I think I just need growth chambers in my office. That seemed to be the key.
So basically, your journey started as a plant scientist, and now you are working on, or your research is one of the applications is this neurodegenerative disorders that you look into. So ALS being one. Which are the other ones that you're looking into? Are there any other ones?
So that's the main focus, but one of the other proteins that also seems to be regulating actin and microtubules. That's sort of where we're going. None of that's published yet, but we're working on it. It causes or gets misregulated in 98% of all neurodegenerative diseases. I read that in a different paper, so someone else said that, but I thought that was really cool. That protein is called TDP43. It doesn't have a very interesting name for that abbreviation, but that protein actually makes these things called biomolecular condensates or liquid phase transitions, which might regulate the cytoskeleton. Some of the early studies on those describing that phase actually did look at the actin cytoskeleton and some of its regulators. So we're sort of looking into, since profilin regulates actin and microtubules, and this thing makes biomolecular condensates, is that a cool way to maybe get your two cytoskeletons to talk to each other? So that's the basic side, but then there's a nice spin to obviously if we see how that's working on a basic level, then what do we do when we have proteins that are harboring the disease mutations, and how does it maybe change those effects?
Okay.
So it's not a secret. They actin and microtubules are regulated somehow by TDP43, and we're sort of going through the data right now to see exactly what the details of that regulation are.
Okay. Wow, that is super cool. So it sounds to me, Jessica, that you're involved, and you have been involved in the past as well, in a lot of cool experiments or research projects. So if you have to pick one research project that you're most proud of, or the most fun or quirky one, could you pick one? I know it's a mean thing to ask, but could you pick one and explain it to us in simple words in the section we call In Other Words?
Yeah, this is not very nice. So I'm proud of everything that we're doing and all the things I've been a part of. And you're right, there's been some really cool things. And I guess I would just say it's probably an outlook thing, right? Everyone, just science is the coolest, just in general. But for your In Other Words section, I guess for right now, the thing I'm the most proud of is this new foray for us into biomolecular condensates. And how is that regulating the cytoskeleton? Because that's sort of really where I think my lab is hopefully going, in a place that we get to use the things that we learned from great people in our past, but also show that we can do it, because I'm still a new PI and, figuring it out.
Okay, so this is like the future thing. So next time you're on the podcast, you will be talking about how you figured it out.
That would be great. I hope we do it.
That sounds amazing. I hope you do. I hope you really do. So Jessica, doing research and publishing, that is like, or doing research in the lab, that is one part of being a scientist or one part of being a professor. There are also other aspects that come along with it. So what else do you like about being a scientist?
Okay, so there's two things, right? I really like helping people just in general. Like, there's not so many things that really make you feel, at least me, feel really good. It's like, if I learn something that I can give to someone else that somehow helps them, that just makes me feel really good. And that's really rewarding to me. So I like helping people. And as a scientist, there's lots of people you can help. Like, you can help students that are like dipping their toe in the science waters, figure out if they think it's interesting. You can help the world because you might be solving something about some puzzle, right? Like, how is this protein doing this thing? Why is that important on a very basic level? Or even like, it could be important for making a drug to treat patients. So, I just like solving problems. So I like helping people and I like solving problems. And, the science ones, there's like not so much drama. You get to learn something that for a moment, no one else knows yet. So that's kind of cool too. Anyway, those are all the things I like about being a scientist.
Okay. Helping people, helping students, and being that first one to know the secret that no one knows before it happens.
Nice. You get like a little secret for a minute. And then it's like, I'm really bad at keeping secrets. So it's like, hey, check out this cool thing.
That is awesome. I think I can't imagine you, from our conversation so far that I know you, I can't imagine you being anything other than a scientist. It's just like, you are like the perfect, we need scientists like you who enjoy the research and science so much.
It's a lot of hard work. So you really have to, be okay with long hours. Sometimes biological things need you,
Yeah,
but,
true.
Yeah.
Absolutely. And speaking of hard work, if you had 3 wishes to improve your research experience, what would you ask for? And I'm not promising anything here, okay?
Okay. So, unlimited microscopes. Not because you need fancy microscopes to do good science. It's just, I believe that more microscopes are always better. It's just, I really love using microscopes. It's really fun. Unlimited funding, because, sometimes, it's sort of nice. Like, when I first got my grant, it was awesome. And sort of, I breathed a sigh of relief because I knew that it was going to be okay. Like, my students in my lab could make some mistakes now, and it wasn't gonna, we were gonna have the money we needed for them to make as many mistakes as they, hopefully not too many, but the mistakes they needed to learn something, but also like move the science forward. And like, just not having to worry about that is sort of that's a pretty big thought. Like, I didn't understand when I was a graduate student or a postdoc, like, yes, funding was important. But when you're sort of responsible for paying people, it makes it a little scarier.
Yeah, definitely.
And then I think the third thing, which, is maybe a little abstract, would be conversations like with all scientists throughout history. Because I just think it'd be really cool. Be like, hey, Aristotle, what do you think about today? What do you think about this problem? Like, couldn't it, like, it would have been cool. Like, Einstein, how would you solve this?
Yeah.
Any of them.
That's a very cool one. We have never had that one. That's a really cool one. That's really, really cool. So, microscopes, unlimited microscopes. Give me your top 3 favorite microscopes, and don't give me this, I like all microscopes and this microscope. No. Top 3 microscopes.
Yeah, that's tricky.
Start with like a 3, 2, 1.
It depends on the experiment.
Okay. Come on.
So I'll say a turf microscope, because that was really the microscope that, like, I got excited about doing microscopy with, and it's really versatile. You can do biochemistry on a cover slip. You could also look at cells. You could also shoot them with lasers.
Oh, what is it? What does TERF stand for? Because I know, I know I'm a material scientist. I know electron microscope, optical microscope. The most I know about biology is 2-photon microscope.
Those are nice too. So TERF stands for total internal reflection fluorescence. So basically, you image a really small part of your sample, about 100 nanometers from the cover slip. And the way you do it is the laser gets reflected back and it makes an evanescent wave. And so it's not really the laser that's like doing it, but it's this evanescent wave. And the evanescent wave goes into your sample and decays exponentially. And the reason that's really cool is you get this really great signal to noise ratio. So like everything looks really crisp and clear, but you only get one like Z plane, if you will. So if there's something like the bottom of the cell or like something on the surface of a cell, it's really great for that.
Okay, TERF microscope.
So TERF, and then some kind of like confocal. I would prefer spinning disc because I have a need for speed. And in dynamics, even though it might not be as pretty of an image, it's always more beautiful because you get more information. I don't know, it has maybe something really high res, like maybe cryo, although I've never done that. But that would be cool. If I could do it again, I would learn more about coding and I would totally do cryo-EM because that would be sweet.
Cryo-EM. Oh my God. We did that during my PhD. I did that. I had like a heart cell on like a recording device and we did like a cross section to see how the heart cell, the cardiomyocyte was sitting on the electrode from which we recorded activity. It was so cool. Oh my God.
That's amazing.
That was so cool. Oh my God, it was so cool. Okay, I just realized that the podcast is called Under the Microscope and we ended up talking so much about microscope. I love this. I love this. So I expect that there would be lots of beautiful images, lots of microscope talk, lots of cups, mugs, for which people have to watch the YouTube video or come on the Twitter account when you're tweeting. So do you want to show your cup?
Oh, my cup. Sorry. This is my narwhal cup that its horn gets like the direction of the twist gets dictated by actin and microtubules, the proteins that I will be talking a lot about when I'm taking over the Twitter.
And speaking of which, what can the followers expect in the week that you are taking over the Real Scientist Nano Twitter account?
So I'm going to start with an intro, just a little bit about me and the lab and how I got here. So those of you that have heard this, spoiler alert, you know some of it. Then I'm going to tell you about some of the open questions in the field that we find really interesting. By no means, I mean, I would love to say we're going to solve all of them, but just to give you a flavor of like what I think is really cool questions that maybe we can help to answer. Then what we're doing to try to address some of those questions. And that involves some pretty cool microscopes and pictures of things from microscopes. So there'll be lots of that. And then some in-depth analysis of how we take one of those images or movies and try to analyze them. And then we're going to wrap it up with like the questions that we hope, like what questions that we didn't solve. But, you know, as you guys know, like a lot of times when you do research, you end up with more questions or different questions than you started with. So we'll talk a little bit about those.
Okay. Wow. So that's quite an action-packed week. Can't wait to have you on Real Scientist Nano. This has been lovely, Jessica. Thank you very much for speaking with me.
No problem. Thank you for inviting me.
Thank you for listening. I also want to thank my team, Johanna, who is our news editor and curator liaison, Swenya, who is our podcast engineer and curator liaison, Lucia, who is our media and website coordinator, and myself, Pranoti, being the owner of the Science Talk and host of Under the Microscope. To know more about us, visit our website, thesciencetalk.com, and follow us on Twitter at realsci_nano.